Hello students and my dear friends, myself Madhu Vancha, Associate Professor of Microbiology. In this session, we'll discuss some of the important microbiology MCQs which will be helpful in the lab technician recruitment examination. First question, unaided human eye has resolution power of option A 0.2 micrometer, option B 0.5 mm, option C 0.2 mm, option D 0.5 micrometer. Here if you see what is meant by resolution power, it is the ability of a lens system to distinguish two objects as a separator rather than one. So resolution power, it is the ability of lens system. So it distinguish between the two objects as a separator. That is called as the resolution power. So unaided human eye, it has resolution power of like a 0.2 mm. The right answer is option C. Here it has a human eye as unaided like a resolution power of 0.2 mm or 200 micrometer. Next question, pox viruses show option A icosahedral symmetry, option B helical symmetry, option C complex symmetry, option D none of the above. Here the right answer is option C complex symmetry. So pox viruses, they do not possess either icosahedral symmetry or helical symmetry. So they possess the complex symmetry. Next question, cuneiform arrangement is characteristic of option A Corynebacterium, option B Bacillus anthracis, option C Clostridium tetani, option D Haemophilus. So here cuneiform arrangement. So here the right answer is option A Corynebacterium. So Corynebacterium diphtheriae, they appeared as like a apple. So after Albert staining, they appears as apple green colored bacilli with the bluish black metachromatic granules. They are arranged in the Chinese letter pattern or cuneiform arrangement. Here the right answer is option A. Next question, Leunston-Jensen media, that is LJ media, it is sterilized by option A inspection, option B autoclaving, option C steaming, option D filtration. So what is LJ media? What is Leunston-Jensen media? So Leunston-Jensen media, it is a selective medium for Mycobacterium tuberculosis. It allows the growth of only Mycobacterium tuberculosis. It is an egg-based medium. So it is an egg-based medium. So this is the only solid media which is prepared without adding agar-agar. So here egg act as a solidifying agent. But the egg, the nature of egg is destroyed at higher temperature. So to avoid that thing, the LJ medium, it is sterilized by the process called as inspection. So in the inspection, like how much temperature is maintained? That is 80 to 85 degrees centigrade temperature is maintained for 30 minutes for 3 successive days. So which means on day 1, we heat the culture media at 80 to 85 degrees centigrade for 30 minutes and we'll keep it aside. On second day also, we'll heat it at 80 to 85 degrees centigrade for 30 minutes. And day 3 also, we heat them at 80 to 85 degrees centigrade for 30 minutes. So by this, we can kill all the forms of the vegetative bacteria as well as the spores. Next question, which one is not a technique for bacterial solid culture? Option A, lawn culture. Option B, streak culture. Option C, stab culture. Option D, none of the above. So if you see the options, lawn culture, streak culture, and stab culture, they are solid cultures. So here the right answer is none of the above. So none of the above is the correct answer. So if you see the other options, lawn culture, stab culture, and streak culture, they are solid culture methods. Next question, which of the following bacteria is an indicator of Anaerobiasis. Option A, Pseudomonas. Option B, Staphylococcus aureus. Option C, Clostridia. Option D, none of the above. So here the right answer is option A, Pseudomonas. So generally in the anaerobic methods of cultivation, so we use biological methods of anaerobic cultures. So in the biological methods of anaerobic cultures, we incubate one aerobic organism, that is strict aerobic organism along with the anaerobic bacteria. So here two blood agar plates. So one is inoculated with Pseudomonas aeruginosa, that is a obligate aerobe. And another is with the like a specimen, so which is expected to be the anaerobic organism. They are incubated like a together, so that this Pseudomonas bacteria absorb the like all the oxygen. So after that, there is no oxygen left for anaerobic bacteria. So, but this method is very slow and ineffective method. Now we are not using it routinely. Next question. Drug of choice for MRSA. What is MRSA? Methicillin resistant Staphylococcus aureus. So which drug is used for MRSA infection? Option A, vancomycin. Option B, penicillin. Option C, cephalosporin. Option D, quinolones. So here in case of MRSA infections, we use vancomycin. So vancomycin is used in case of MRSA infection. It is a bactericidal drug. It kills the bacteria. Next question. Which of the following is an automated continuous monitoring system of blood culture? So automated continuous blood cultures. Option A, BACT or Alert 3D. Option B, BACTEC. Option C, Versatrec. Option D, all of the above. Here, the right answer is option D, all of the above. If you see BACT, Alert or Alert 3D or BACTEC or Versatrec, all these are automated methods. So if you see the first one, Alert 3D, so what is the principle of this one? It is based upon the calorimetric detection of growth of organism. It is based upon calorimetric detection of growth of organism. And if you see BACTEC, it is based upon the detection of fluorescence. So whenever the organism grows in the culture media, based upon the fluorescence, that will identify the growth. Next, Versatrec. So it detects the mycobacterial growth automatically. It will detect mycobacterial growth automatically by monitoring the rate of oxygen consumption. By monitoring the rate of oxygen consumption within the headspace of the culture bottle for every 24 minutes. So here the right answer is option D, all of the above. Next question. Transport media used for Vibrio cholera in stool. So what is transport media? Transport media, it do not allow the bacteria to multiply, but it maintains the viability. Option A, VR medium, that is Venkatraman Ramakrishnan medium. Option B, autoclaved seawater. Option C, Kerry-Blair medium. And option D, all of the above. Here the right answer is option D, all of the above. If you see Venkatraman Ramakrishnan medium, autoclaved seawater, and Kerry-Blair medium, these three culture medias are transport medium. They do not allow the bacteria to multiply, and they maintain the viability. They do not allow the bacteria to multiply, but they maintain the viability of the cells. Next question. Antibiotic sensitivity AST for Streptococcus pneumoniae is done on option A, Muller-Hinton agar medium. Option B, Muller-Hinton agar with 5% sheep blood. Option C, nutrient agar. And option D, none of the above. So if you see these options, so generally for antibiotic sensitivity test, we use Muller-Hinton agar medium. And alternative to Muller-Hinton agar medium is nutrient agar medium. So generally we use Muller-Hinton agar medium, and alternative to Muller-Hinton, that is nutrient agar. But in case of Streptococcus pneumoniae, it is a fastidious organism. So what is fastidious organism? Which is exact in their growth requirement. So in that cases, like we add 5% of sheep blood. So to the normal like Muller-Hinton agar only we add 5% sheep blood. So here the right answer is option B, Muller-Hinton agar with 5% sheep blood. Next question. Diameter of a antibiotic disc used in antimicrobial sensitivity testing is. So what is the diameter of antibiotic disc? Option A 6 mm, option B 5 mm, option C 3 mm, and option D none of the above. If you see, the right answer is option A 6 mm. So the filter paper disc which is having 6 mm diameter is used. So for preparation of antibiotic discs, pure antimicrobial agents should be used. So we are not supposed to use the antibiotics which are intended for clinical purpose. Next question. MIC of an antibiotic can be detected by option A, broth dilation method, option B, epsilometer test, option C, both A and B, option D, none. Here what is MIC? MIC means a minimum inhibitory concentration. So what is minimum inhibitory concentration? That is the lowest concentration of the drug at which there is no visible growth. Here the right answer is option C, both A and B. That is broth dilution method and epsilometer test. They detect the MIC of an antibiotic. Next question. All are methods of antimicrobial susceptibility testing except option A, E-test, that is epsilometer test. Option B, Kirby-Bar disc diffusion method. Option C, Stokes disc diffusion method. And option D, moldy TOF. So here the right answer is option D. So if you see epsilometer test, Kirby-Bar disc diffusion method, Stokes disc diffusion method, these three are antimicrobial susceptibility test. So if you see, that is Manley-Topf, that is matrix assisted laser desorption or ionization time of flight. TYF means time of flight. So this is used for rapid identification of bacteria, fungi, and mycobacteria with absolute accuracy. So remaining three, they are used for antibiotic susceptibility testing. And this is for identification of bacteria. The moldy TOF, it is for identification of bacteria, fungi, and mycobacteria. Next, three components of PCR, that is polymerase chain reaction, involves all except option A, DNA extraction, option B, amplification, option C, gel documentation, option D, blotting. Here the right answer is option D. So in the PCR, blotting step is not involved. Blotting is not the component of PCR. Next question. Which of the following is not an ingredient of basal medium? So what is basal medium? Basal medium is like a simple medium or basal medium, which is having like basic ingredients. Option A, blood. Option B, agar. Option C, NaCl, sodium chloride. Option D, peptone. Here the right answer is option A, blood. So if you see nutrient agar or nutrient broth is the basal medium in microbiology. So nutrient agar or nutrient broth, it consists of agar. So if you say nutrient agar, it consists of agar, sodium chloride, and peptone. So blood, it is added only in the enriched media. So to the basal media, if we add blood, it will become the enriched media. Next question. Free DNA can be transferred between bacteria by option A, transformation, option B, conjugation, option C, transduction, option D, transposition. Here the right answer is transformation. So what is meant by transformation? That is the transfer of free DNA or naked DNA. And if you see other option, conjugation. What is conjugation? It is the process of transfer of DNA from F plus cell to the F minus cell. So that is genetic material is transferred from F plus cell to the F minus cell. So that is with the help of the pili, which is with the like a sex pili. So sex pili, with the help of the sex pili, F plus cell attaches to the F minus cell and they form the conjugation tube. And if you see transduction, so what is transduction? In the transduction, virus, it transfers the genetic material from one bacterium to the another bacterium. That is by the bacteriophages. Next, transposition or transposition. So it is a segment of DNA that has ability to move around between chromosomal and extra chromosomal DNA molecules within the cells. Next question. Methicillin resistant in Staphylococcus aureus is mediated by option A, MEC-A gene, option B, plasmid, option C, transposons, option D, none of the above. Here the right answer is option A, MEC-A gene. So this gene is responsible for like a methicillin resistance in Staph aureus. Next question. Obligate intracellular bacteria are all except. What is obligate intracellular? Obligate means strict. They are strictly present inside of the cells. Option A, Mycobacterium leprae. Option B, Rickettsia. Option C, Chlamydia. And option D, Mycobacterium tuberculosis. So here the right answer is option D, Mycobacterium tuberculosis. So except this Mycobacterium tuberculosis, remaining three organisms are obligate intracellular bacteria. They are strictly present inside of the cells. Next question. Following bacteria does not require large infective dose. So that is MID, minimum infective dose, which means minimum number of bacteria required to produce the clinical infection. So here option A, Shigella, option B, E. coli, option C, Salmonella, option D, Brucella. Here the right answer is option A, Shigella. So here the Shigella, it has like a very less infective dose, that even 10 to 100 bacilli, even 10 bacilli, 10 to 100 bacilli also can cause the infection because they can tolerate the gastric acidity. Next question. All are non-enveloped DNA viruses except option A, parvovirus, option B, adenoviruses, option C, papovavirus, option D, heparinavirus. So here the right answer is option D, heparinavirus. So heparinavirus, they include hepatitis B virus. So it include the hepatitis B virus. Hepatitis B virus is enveloped viruses. If you see other options, parvovirus, adeno, and papovavirus, they are all non-enveloped DNA viruses. They consist of DNA as genetic material. Next question. Which of the following protozoa has two hosts in its life cycle? Option A, entamoeba. Option B, giardia. Option C, malarial parasite. Option D, trichomonas. So which of the following organism has two hosts? So here the right answer is option C, malarial parasites. So malarial parasite, that is Plasmodium, it completes its life cycle in two hosts. One is definitive host and another is the intermediate host. So what is definitive host? In which host body the sexual stages of parasite takes place, that is called as the definitive host. And what is like an intermediate host? So in which host body the asexual stages of parasite takes place, that is called as the intermediate host. So in case of malarial parasite, the sexual stages of parasite takes place in the mosquito. So mosquito is the definitive host. And the asexual stages of parasite takes place in the humans. So humans are intermediate hosts. So if you see other options such as entamoeba, giardia, and trichomonas, they complete their life cycle only in one host, that is in the humans. Next question. Which of the following trematode is transmitted by skin penetration? Option A, Phaeola. Option C, Clonarchia sinensis. Option C, Cystosoma. Option D, Paragonemias. So here the right answer is option C, Cystosoma. So here the Cercaria, that is like an infective form of the like a Cystosoma. So it can penetrate the like unbroken skin. So it is like even though you have no cuts on the like skin, so this Cercaria of cystosoma, it can penetrate the like skin and it can enter into the host body and can cause the infection. Next question, all are yeasts are yeast-like fungi except option A, Candida, option B, Trichophyton, option C, Cryptococcus, option D, Trichospora. Here the right answer is option B, Trichophyton. Trichophyton, it is not a yeast, not yeast-like fungi. It belongs to the dermatophytes, which causes skin infections. Next question, the pH of SDA medium, that is Sabouraud's dextrose agar, should be. So Sabouraud's dextrose agar, so that is the culture medium which is used for growing up the fungi. So what is the pH of SDA medium? Option A, acidic. Option B, basic. Option C, neutral. Option D, both A and C. Here the right answer is option A. So SDA, it has slight acidic pH. So what is the pH of SDA? That is 5.0. Next question, all are fungal stains except option A, chalcophlorite, option B, periodic acid stain, option C, Meyer's musicaramine, and option D, Ponders. So here the right answer is option D, Ponders. So if you see option A, chalcophlorite, periodic acid stain, Meyer's musicaramine, so these three stains, they helps to identify the fungi. They are fungal stains. So if you see Ponders stain, so it is helps to identify the Corynebacterium diphtheria. Here the right answer is option D. Next question, aerosol transmission is prevented by option A, surgical mask, option B, N95 mask, option C, gown, option D, none of the above. So aerosol transmission means they are smaller droplets. So smaller droplets which are less than 5 micrometer in the diameter. So the smaller droplets which are less than 5 micrometer in the diameter, they cannot transmit beyond 1 meter. So how they can be prevented? By wearing the N95 mask. Next question, time taken for hand scrub is option A, 5 to 7 minutes, option B, 1 minute, option C, 1 second, option D, 15 minutes. Here the right answer is option A, 5 to 7 minutes. So what is the time for hand scrub? That is minimum 5 to 7 minutes. Next question, disinfectant used to kill all the spores is option A, chlorhexidine, option B, triclosan, option C, hypochlorite, option D, 5% phenol. So here the right answer is option C, hypochlorite. So this is a disinfectant which kills the like spores. So that is one of the high level disinfectant. Next question, screening for nasal carriage of bacteria. Which bacteria is done in healthcare workers? Option A, MRSA, option B, Clostridia, option C, Lactobacillus, option D, Bacillus species. Here the right answer is MRSA. So MRSA is screening is done in case of healthcare workers. Next, HEPA filter. So what is mean by HEPA filters? High efficiency particulate air. High efficiency particulate air. So they filters up to the high efficiency filters filter particles up to option A, 4 micrometer, option B, 0.3 micrometer, option C, 1 nanometer, option D, 5 micrometer. Here the right answer is option B, 0.3 micrometers. So HEPA filters, they are fixed in the laminar airflow as well as in some of the like cubicles. So what they do, they deliver the like a clean free bacterial, like a clean free bacterial like air. So here it can filter the particles up to the 0.3 micrometer. Next question, healthcare workers, they should be vaccinated against. So all the healthcare workers, they must be vaccinated against. Option B, HBV. Option A, HBV, that is hepatitis B virus. Option B, influenza. Option C, typhoid. And option D, all of the above. So here the right answer is all of the above. So for all the healthcare workers, so these vaccines like hepatitis B vaccine, influenza, and typhoid vaccine should be given. Next, positive tuberculin test is an example of option A, type 1 hypersensitivity, option B, type 2, option C, type 3, option D, type 4 hypersensitivity. So positive tuberculin test, what is the answer? Here that is type 4 hypersensitivity, that is delayed type of hypersensitivity or cell-mediated hypersensitivity that belongs to the tuberculin skin test. Next question, anti-citrulline anti-citrullinated peptide antibodies, that is anti-CCP antibodies, are diagnostic for option A, SLE, that is systemic lupus erythematosus, option B, rheumatoid arthritis, option C, Crohn's disease, option D, scleroderma. Here the right answer is rheumatoid arthritis. So anti-CCP antibodies we can see in case of rheumatoid arthritis. So normal range of anti-CCP antibodies, they should be less than 20 EU per ml. So what is EU per ml? Enzyme units per milliliter. So anti-CCP antibodies can be seen in case of rheumatoid arthritis. RA. Next question, quality concepts include option A, quality management system, option B, quality control, option C, quality assurance, option D, all of the above. Here the right answer is option D, all of the above. So quality concepts include quality management system, quality control, quality assurance. QA. Next question, which of the following disinfectant is used for blood spillage area? So here how the spillages are cleaned. Option A, phenol, option B, Lysol, option C, hypochlorite, option D, formaldehyde. So here the right answer is option C, hypochlorite. So freshly prepared 1% sodium hypochlorite is used in case of a spillage area. So the spillage area should be covered with a sodium hypochlorite solution, freshly prepared sodium hypochlorite solution for minimum of 15 to 20 minutes. Next question, culture plates of bacteria are pre-treated by option A, autoclaving, option B, incineration, option C, 1% hypochlorite, option D, washing. So how the culture plates are pre-treated, how the culture plates are freed from the microorganisms. That is done by autoclaving. The right answer is option A, autoclaving. Next question, gloves are discarded in which bag? Option A, yellow, option B, red, option C, black, and option D, blue. Here the right answer is option B, red. So here the gloves, they should be discarded in red color bag or red color container. Next question, which of the following is not a vector-borne disease? So which is not transmitted by vector bite? Option A, malaria, option B, dengue, option C, Kala Azar, option D, salmonella. Here the right answer is option D, salmonella. Okay, so here salmonella, it is not a vector-borne disease. It is transmitted by contaminated food and water. So if you see malaria, dengue, they are transmitted by the bite of the mosquitoes. And if you see Kala Azar, that is transmitted by the sand fly. Next question, booster dose for HBV, hepatitis B vaccine, should be given when anti-HBs AG titer is option A, that is more than 10 IU per ml, international units per ml. Option B, less than 10 international units per ml. Option C, more than 100 international units per ml, and option D, none of the above. Here the right answer is option B. So if anti-HBs AG titer is less than 10 international units per ml, we should give the booster dose again. Next question, transfusion transmitted infections can be? Option A, HBV, option B, HIV, option C, syphilis, and option D, all of the above. Here the right answer is option D, all of the above. So all this hepatitis B virus, HIV, human immunodeficiency virus, and syphilis, they can be transmitted by contaminated blood as well as the blood products, transfusion of contaminated blood and blood products. Next question, laboratory acquired infections can be caused by all except option A, Brucella, option B, Bacteria, option C, Mycobacterium, option D, Lactobacillus. Here the right answer is option D, Lactobacillus. So here if you see Brucella, Bacterium, Mycobacteria, like they are all laboratory acquired infections. Next question, agents that can cause post-transplant infections are all except option A, cytomegalovirus, option B, Toxoplasma, option C, Candida, option D, Entamoeba. Here the right answer is option D, Entamoeba. So Entamoeba cannot cause the post-transplant infections. So remaining they causes the infections. Next question, pH indicator that has alkaline range is option A, phenolphthalein, option B, bromophenyl blue, option C, methyl red, option D, bromocresol green. So here the right answer is option A, phenolphthalein. So phenolphthalein, it has alkaline range, alkaline range, that is 8.0 to 9.0. Next question, preservation of bacteria can be done by option A, 15% glycerol, option B, nutrient agar stabs, option C, lyophilization, and option D, all of the above. So here the right answer is option D, all of the above. So for long-term storage of the bacteria, we can go for 15% glycerol or nutrient agar stabs or lyophilization. Next question, broken glass tube is discarded in which bag? Option A, red, option B, yellow, option C, white, and option D, green. Here the right answer is option C, white. So broken glass materials, they should be discarded in the white bag. Thank you.